Hello, my name is Irith Aloni, and I will tell you about social inheritance as a novel mechanism shaping the genetic structure of populations. So let's start with the definition. Social inheritance refers to the tendency of young animals of some social species to connect with individuals which are connected to the mother or to both parents. Now, in this study, I investigated the question of whether and how social inheritance affects the genetic structure of the population. And in order to assess this question, I used simulation models. My model consists of a population of n individuals, which are randomly sexed, and each one receives a hypothetical set of 10 random genes, where each gene has 2 alleles, and each allele has an initial frequency of 1/2. Now, on each iteration, that is, each cycle of the simulation, one random individual dies, and then a random female is selected, and the random male is selected out of this female's contact group. Now, from this pair of a male and a female, we get a newborn who inherits its genetic material in a standard way, and then it forms social contacts according to 2 fixed probability rules, which we name Pn and Pr, where Pn is a social inheritance probability, that is, a probability to connect with the mother's contacts, and PR is a probability to connect with individuals which are not connected to the mother. Now, such an iteration is repetitively run 10n times to simulate 10 generations, and each combination of parameters, that is, specific values of Pn, Pr, and n, are used in 30 simulations to gain some statistical power. Now, at the end of a simulation, the computer does 3 things. First, it applies an algorithm called walk trap, which detects the group structure of the social network. Secondly, it calculates Wright F values, which are common measures of population genetic structure. And eventually, it calculates relatedness between individuals based on a pedigree that is recorded along the simulation. Now, Wright's F values include 3 types. We have FST, which is the most important one from my perspective, as it measures genetic distance between groups. FIT measures the overall deviation of the population from Hardy-Weinberg equilibrium, and FIS is the average deviation from Hardy-Weinberg within groups. Now, under random circumstances, all F values would be 0, and an F value larger than 0.1 is considered an indication for some deviance from equilibrium. So let's look at the results. Here we see box plots of Wright's F values for the different simulations. On the top line, you can see the different Pn values I have used. On the side here, you see K, which is the network degree, that is the average contact group size. And the different colors stand for the different F values, F types. And you can see that FST, which, as I said, is a measure of genetic distance between groups, is very high. FST and also FIT are very high when the Pn levels are high and the network degree is relatively low, which means that high social inheritance is associated with considerable genetic differences between social groups. Now, additionally, you may notice that FIS are all very low in pink here, which means that we have a random mating within groups. Here you see relatedness within contact groups, and you may notice that we get a very similar pattern to what we saw above. Relatedness values get high when Pn values, social inheritance levels, are high, and degree is relatively low, which means that under high social inheritance levels, relatives stay connected, which makes perfect sense. However, this high level of relatedness made me suspect that maybe some random inbreeding is going on, which affects the patterns that we see. So in order to test for it, I run another set of simulations in which I prevented mating between first-degree relatives. And as you can see, the pattern here is almost identical to what we saw above, and so is relatedness. Now, finally, I wanted to check how dispersal would affect the pattern. So in order to simulate dispersal, I changed the rules for the males, and now a newborn male did not follow anymore its mother's contacts, but instead newborn males were assigned a random individual which served as a fostering parent, so to speak, and the newborn would apply the PNPR rules to the fostering parent's contacts. And as you can see, the pattern in this case collapses completely. Values are essentially zero. Look at the scale, and we really have a random mating system here. When we look at relatedness, well, we still see a pattern, but look at the scale. Again, values are very low and insignificant. And in fact, this was in a way even surprising, because if you think about it, females still keep following the regular rules, following the mother's contacts when forming connections. And you would expect that under high levels of PN, high levels of social inheritance at least, we'll get matrilines. And therefore I expected to see somewhat higher relatedness levels, but we don't. So, to sum up, I have shown here that social inheritance is associated with population genetic structure. I found considerable genetic differences between social groups and the high levels of social inheritance. I also found that under high levels of social inheritance, relatives tend to cluster, and male dispersal has prevented the formation of any genetic differences between groups. It kept mating completely random and relatedness levels low. And thank you for listening.